Regulation of Adaptive Behaviour During Fasting by Hypothalamic Foxa2 by Jose P. Silvart et al. Presented by Louise Elliott.
Alistair Shaw.
Ashley Elliott.
Introduction.
This study, taken from issue 7273, volume 462 of Nature, is aimed towards discovering the mechanism of action that links insulin, Foxa2, and melanin-concentrating hormone and orexin.
The lateral hypothalamic area is considered a classic feeding centre, regulating food intake, orexin and melanin-concentrating hormone. These neuropeptides are inhibited in response to feeding-related signals and are released during fasting.
Fox-ahead Fox transcription factor, Foxa2, a downstream target of insulin signaling, regulates the expression of orexin and MCH. During fasting, Foxa2 binds to the MCH and orexin promoters and stimulates their expression. In fed and hyperinsulinic obese mice, insulin signaling leads to nuclear exclusion of Foxa2 and reduces expression of MCH and orexin.
The question being asked by the study is whether insulin directly affects Foxa2 expression or Foxa2 location. Introduction. The proteins. Foxa2 encodes for a member of the human fork head class of DNA binding proteins. These are nucleotide factors that are transcriptional activators for various different genes. Such genes include albumin and, in this case, MCH and orexin. Similar family members have been found in all mammals, so some mouse models can be used in this study. This transcription factor has been implicated in being a morphogen. It is a metabolism regulator, regulating the transcription of the enzymes for beta oxidase and ketogenesis.
MCH and orexin are short neuropeptides that are released from the LHA.
MHC has been linked to changes in behaviour, making the subject irascible and highly motivated to find food under starvation.
Insulin is a signal protein that changes intercellular conditions to allow glucose into tissues such as adipose and skeletal muscle. It does this via a surface receptor that phosphorylates tyrosine residues in molecules such as insulin receptor substrate 1.
Tissue types. The lateral hypothalamic area is two areas of the brain, one in each hemisphere, that controls feeding in mammals. Although it is relatively small, its axons are spread throughout the cortex and therefore create a system-wide behavioural change when activated.
MCH neurons are located in the anterior part of the LHA. Orexin neurons are predominantly found in the posterior part of the LHA, the zona incerta, and the subthalamic nucleus.
Methodology.
Mice were starved or fed in the preceding 24 hours to the experiment. 1 picogram human insulin or PBS was injected to the study mice's tail veins. PBS is phosphate buffered saline, which is a buffer solution. It is water-based salt solution containing sodium chloride, sodium phosphate, and in some formulations potassium chloride and potassium phosphate. The buffer's phosphate group helps maintain a constant pH. It was used as a control. The mice were killed after 20 minutes and the brains were processed into 200 micrometer slices in the regions of the lateral hippocampus that have MHC and orexin expressed. This is to allow staining to occur.
Foxa2 was mapped using fluorescently tagged antibodies. Cultured hypothalamus from the mice was placed for 4 hours in media containing either a low 5 millimolar or a high 16 millimolar concentration of glucose in the absence of insulin, or a low glucose concentration in the presence of 100 nanomolar insulin. Western blotting was done to show relative concentrations and therefore position of Foxa2 in the nuclear and cytosolic extracts of LHA.
Results.
This image shows brain sections of fed mice that were stained with antibodies against Foxa2 and orexin, or against Foxa2 and MCH. These were then counterstained with the nucleodide DAPI, which is a fluorescent stain that binds strongly to AT-rich regions in DNA. arrows indicate neurons in which both Foxa2 and orexin co-localize.
This image shows that Foxa2 and the non-insulin dependent Foxa1 were discovered in both MCH and orexin neuronal clusters.
The Western blot shows that Foxa2's location, not its expression, is dependent on insulin. The immunoblotted nuclear and cytoplasmic hypothalamic extracts of fasted and fed wild-type mice show that when fed, Foxa2 is purely systolic, but under starvation it is nuclear. So Foxa2 is nuclear with glucose and cytosolic with insulin. Detection of the TATA binding protein TBP was used as a loading control. The TATA binding protein is a general transcription factor that binds specifically to a DNA sequence called a TATA box.
Co-localization of Foxa2 with orexin or MCH is demonstrated by the yellow staining of neurons. Immunofluorescence analysis is done to show the subcellular distribution of Foxa1 and Foxa2, and this was performed in fasting mice following the tail vein injection with either insulin or PBS. This reinforced the Western blot of the nuclear and cytosolic extracts by showing visually the positions of Foxa2 in insulin-affected LHA.
Sections were stained with antibodies against Foxa1 or Foxa2 and counterstained with the API. Immunohistochemical analysis of hypothalamic sections of fasted and fed mice stained with anti-Foxa2 antibodies and the API. This also supports the Western blot by showing the position of the nucleus fluorescently. Conclusion.
In conclusion, the evidence suggests that Foxa2 expression is not affected by insulin, therefore it is affected by an intermediate molecule phosphorylating Foxa2. Phosphorylation changes the intracellular targeting, and this change prevents Foxa2 entering the nucleus and preventing it operating on its targets, which are the promoters for MCH and orexin.
This then prevents MCH and orexin performing the system-wide behavioural changes in this case mice, preventing them from being hungry and searching for food.
To help prove that Foxa2 affects neuropeptide production, a study could be done that genetically modifies mice to overproduce Foxa2 to observe any behavioural changes that occur.
Thank you for watching.